Okay, so today we're going to look at eukaryotic cells and their exchange surfaces and their organelles. So there's two types of cells, prokaryotic and eukaryotic. We'll spend another session at some point looking at prokaryotic. I'm just going to look at eukaryotic today. So in the past, especially like at GCSE, you sort of get shown a cell, and I've probably got an image of one on here. Here we go, a GCSE typical plant and animal cell. So we go, okay, they're a eukaryotic cell. So a eukaryotic organism is an organism that's made up of multiple cells. So, and we would have these typical basic structures: an animal cell that's got nucleus, cytoplasm, cell membrane. Sometimes we'd have on there ribosomes and mitochondria. And we would have a plant cell with the extras of the chloroplast, vacuole, and cell wall. And that sort of as complex as we'd go. But actually, eukaryotic cells are really, really complex. So what we're going to look at now is the actual complexity of all the organelles that are there and fully what each of the organelles does. So we're going to look into them in a lot more detail. So yeah, we're going to look at eukaryotic cells first, look into all the different details of them, and then we'll have another lesson where we look at prokaryotic cells. So eukaryotic cells in general are a lot more complicated than prokaryotic cells. In general, you might get the odd prokaryotic cell that's a bit more complex. But obviously, prokaryotic cells are prokaryotic organisms. They're single-celled organisms. So yeah, our eukaryotic cells are a lot more complex. So we've been looking at these images for years. We're probably generally familiar with some bits, and like I said, got an animal cell there, the really basic point. That's probably more of a key stage 3 diagram. GCSE, we would have some little dots on there for ribosomes, and then some little semicircle-y, oblong-y things for mitochondria. And that's sort of all we sort of know. And plant cells, again, when we add all these extra organelles into our animal cell that we're about to go into in a minute, plant cells have exactly the same as animal cells, plus those extras that we've spoken about in the past. So they still have that, obviously they have that cellulose cell wall. They have something called a plasmodesmata, which we'll look into in a bit more detail. Still the vacuole and the chloroplasts. So all these extra organelles that we're going to talk about in a lot more detail, and we'll probably have it with an image of an animal cell. These all also apply to a plant cell, so that's the important part to think about. But those plant cells obviously have those extra parts. Eukaryotic cells are, we generally talk about them being animal and plant cells. They are also the cells that are found in algae and fungi. So algal cells look a lot like plant cells. They have exactly the same organelles, including a cell wall and chloroplast. And that's because algae carry out photosynthesis like plants, but they can be single-celled or multicellular. So they're classed as a eukaryotic cell, even though they could actually be single-celled organisms. Fungal cells are also very similar to plant cells, but they have two key differences. Their cell walls aren't made of cellulose, they're made of chitin, and they don't have chloroplasts because they don't photosynthesize. So our fungal cells would make up organisms such as mushrooms and yeast. So cells. Cells all have an internal structure, and this internal structure we talk about is called the ultrastructure. And as we've said, we've got eukaryotic cells and prokaryotic cells. From Greek, eukaryotic means true nucleus, and prokaryotic means not having a true nucleus. So eukaryotic cells are defined as having a distinct nucleus, and they possess a membrane-bound organelles, and epithelial cells are concerned with absorption and secretion. So eukaryotic cells, we're going to look at the nucleus Yes, we are. We'll look at the nucleus to begin with, because actually in the past at GCSE, we've generally just had like a picture of a nucleus. So we've sort of drawn a cell with a black dot in it and gone, yep, that's a nucleus. It's got DNA in it, controls the cell, controls the organism. And that's sort of where we sort of ended with any detail and any specific information about the nucleus. So what we're going to look at now is really, really actually further detail. So a nucleus isn't just a nucleus. It's not just that. It's not just this thing that has DNA in it. Or actually, it's made up of multiple different layers. So the nucleus contains a nuclear envelope, and the nuclear envelope is the outer layer of the nucleus. So if we have a look at this image here, the nuclear envelope is this whole outer layer. And again, when we talk about cells, we have to remind ourselves that cells are these 3D objects. It's like an egg that we've then gone and cut in half and we're looking into, so we're looking down on it. So this image is quite nice because we've done taken a quarter of it out, so we're looking at it so we can still see its 3D shape, and that's obviously a nucleus. That's not a whole cell. And so actually the nucleus of the cell is also another round spherical structure inside the cell. So inside the cell we then have the nucleus, which is its own spherical structure, and what's going to cover the outside of the nucleus is this nuclear envelope. So it's a nuclear envelope, and a nuclear envelope is a double membrane that controls material entry and exit in a very similar way to the cell surface membrane of the actual cell. So the nuclear envelope is a double membrane similar to our cell surface membrane, and it controls material entry in and out of the nucleus. The nuclear envelope has nuclear pores in it, and these are what allow large molecules like mRNA out of the nucleus. So the nuclear envelope controls what's going in and out, very similar to our membrane. Some things will be able to diffuse in, diffuse out. mRNA is too large a molecule to just diffuse out, so we have these pores. And these pores, so again, if we look at this image here, these little circles here that we're pointing at, these pores are how the mRNA leaves the nucleus. Leaves the nucleus. So yeah, those nuclear pores, they're what allow our mRNA to actually go out and leave our nucleus. The nucleus itself contains chromosomes, which are made from protein-bound linear DNA, often called chromatin, which is made of proteins and DNA, controls the cell activity. And the nucleus contains chromosomes, and we call this, and one more structure, which we call the nucleolus. The nucleolus is the area of the nucleus that makes ribosomes. So we always talk about ribosomes being in the cell. We never really talk about where ribosomes are made. They're actually also made in the nucleus. The plural of nucleus is nuclei, and the plural of nucleolus is nucleoli. So it's a little bit weird. So nucleolus is the singular, and nucleoli is the plural for nucleolus. Nucleus is the singular, nuclei is the plural. So the nucleus, the whole job of it is to control the cell's activity. And it does this by controlling the transcription of DNA. It's basically the production of proteins and protein synthesis, which we will look at a later date. DNA contains our instructions to make our proteins, and the pores, those nuclear pores, allow substances such as RNA to move between the nucleus and the cytoplasm, and the nucleolus makes the ribosomes. So if we look at this image here, so our nucleus functions are produce mRNA for protein synthesis. They retain genetic material in the form of DNA or chromosomes, and they manufacture ribosomal RNA and ribosomes. So when we look at it now, this is actually our structure. We have this outer layer, which is our nuclear envelope. Sometimes it's called a nuclear membrane, but officially it's a nuclear envelope. We've got our nuclear pores. We have our chromatin, And our chromatin is our area where we have all the different genetic information. We have our nucleolus, which actually has the DNA inside it. And then we've got our different membrane areas. So that's the nucleus. What we're going to have a look at now is the mitochondria. Now, you'll probably notice that I'm going to miss out the cell surface membrane of a cell. The main reason I'm going to miss out cell surface membrane is this becomes basically a topic of its own because it's so complex. At this very moment in time, all we need to know about cell surface membrane is very similar to what you knew at GCSE. It's a membrane found on the surface of animal cells and just inside the cell wall of other cells, and it's made of lipids and proteins, and it regulates the movement of substances into and out of the cell. It also has receptor molecules on it which allow it to respond to chemicals like hormones. So it's just this layer that controls what comes in and out of the cell, and that's all we need to know right now. So we're going to look into mitochondria now in a little bit more detail. So this is what a mitochondria actually looks like, and we have to understand the actual structure of these organelles. It's why I had that image of the nucleus here. We need to actually know the structure of the nucleus. So now we need to understand the structure of mitochondria. So mitochondria are the main organelles involved in respiration. Respiration is something that I can't remember off the top of my head, I think it's second year, probably, I think it's A2, where you do something called Krebs cycle oxidative phosphorylation, where we look into respiration in a lot more detail. But mitochondria are responsible for production of the energy carrier molecule ATP. So that ATP that we spoke about last session, mitochondria is the site of production of that energy carrier molecule ATP. They are an oval shape and they have a double membrane. This double membrane, they have an inner and an outer membrane. The inner membrane is folded to form structures called cristae, and these are extensions of the inner membrane. And basically the point of these is to increase the surface area for attachment of enzymes. And inside the inner membrane, we find the matrix. Now the matrix is involved in is semi-rigid, and it contains proteins, lipids, and trace DNA. It's the active site of the Krebs cycle, so it's actually the active site where respiration occurs. Right now, we can class it as a site of the mitochondria where respiration happens. Later on, when we go into respiration, it's specifically where the Krebs cycle happens. So the function of the mitochondria is that it is the site of aerobic respiration. It is also where ATP is produced, and they're found in large numbers in cells that are very active and require a lot of energy. Again, a plural bit of information here. A single mitochondria is mitochondria. And the plural of is, right, let me have a word that. Mitochondria is the plural, the singular is mitochondrion. Just a little bit of information there about it. And then we go on to the endoplasmic reticulum. Now, this is one that you probably won't have heard of really at all. So the endoplasmic reticulum, when we look at it on that image there, it's actually, as you can see, it's joined to the nucleus. So it's a system of sheet-like membranes that are found throughout the cytoplasm. So we often talk about the cytoplasm at GCSE as just being this jelly-like substance that fills the whole of the empty space in the cell. Actually, we have this endoplasmic reticulum in there, and it is continuous with the outer nuclear membrane. So it is attached to the outer nuclear membrane, the outer nuclear envelope, and it is continuous folds. Now we have two types of endoplasmic reticulum. We have rough ER, rough endoplasmic reticulum, which has ribosomes present on its surface, and we have smooth endoplasmic reticulum where there is no ribosomes. So our rough ER, we can tell the difference of it is just because on a drawing, if we have an image of it here, yeah, here we go. We pop little dots on for the ribosomes, so it's rough because it's got ribosomes present. Smooth doesn't have dots because it doesn't have ribosomes present. So rough endoplasmic reticulum, it's covered with ribosomes. It's got a large surface area for protein synthesis, which makes sense because we talk about ribosomes being the site of protein synthesis. And it is a pathway to transport proteins out of our cells. Whereas our smooth ER carries out, oops, what's happened here? Our smooth ER carries out a different job. Because there is no ribosomes present, it is a site of synthesis and storing and transporting of lipids, and it's also the site of synthesis, storing and transporting carbohydrates. And so there's the images of them again. They do look quite different, but they do both carry out very different jobs. Then we have the Golgi apparatus or apparatus, sometimes called the Golgi complex. It's made up of two parts. So we have the Golgi apparatus and the Golgi vesicles, which we'll look into a bit more detail. So the Golgi apparatus, its structure is very, very similar to smooth endoplasmic reticulum, but it's just a lot more compact. So it's just squished in basically a lot more. And it's basically flattened stacks of membranes with rounded structures called vesicles. And we'll look into those vesicles in a minute. The job of this is it processes and packages new lipids and proteins, and it also makes something called lysosomes. So proteins and lipids are produced by the rough endoplasmic reticulum, and they pass through the Golgi apparatus to be modified. So here they're being processed, they're being packaged. Once sorted, proteins and lipids are then transported into our vesicles that fuse with the cell surface membrane to release their contents. So we make the proteins and lipids. Well, we make the proteins in our rough ER, our lipids in our smooth ER. They pass through the Golgi to be modified. So they're packaged and processed, and then they're put into the vesicles, which then these vesicles travel to the cell membrane, and they fuse with the cell surface membrane to release their contents. So that's how things like proteins and lipids leave the actual cell. And it's through a process called exocytosis. Rather than like diffusion through the cell membrane. And so this is what Golgi apparatus looks like. Again, you can see how it does look very similar to the smooth endoplasmic reticulum, and we have these membrane-bound vesicles. So the Golgi, its functions, it adds carbohydrates to proteins to make them glycoproteins. It produces secretory enzymes. It secretes carbohydrates. It transports and modifies lipids. It also forms lysosomes. Lysosomes, I thought I had something on the vesicles here. Right, let me just go back to my Golgi for a minute. So those Golgi vesicles that we talk about, so we've got one of them here. These are small fluid-filled sacs in the cytoplasm. So they're not technically attached to the Golgi apparatus. They're sort of to the side of it in the cytoplasm. And they're surrounded by a membrane, and they are produced by the Golgi apparatus. What they do is they store lipids and proteins that are that have passed through the Golgi and transport them out of the cell via the cell surface membrane. So these are like the transporters. They carry proteins and lipids from the Golgi all the way to that cell surface membrane. And we've said Golgi can make lysosomes. So here is a lysosome. So lysosomes are formed in the Golgi, and they contain enzymes. Lysosomes are round, and they're surrounded by a membrane with no clear internal structure. We actually say they're a type of Golgi vesicle. And what they do is they contain digestive enzymes called lysosomes. The only difference is lysozymes has a Z in it, lysosomes is a S, so it's lysozymes. So what they do is they isolate these potentially harmful enzymes from the rest of the cell. These enzymes are digestive enzymes. Because they are digestive enzymes, and if you think of all the different components of our cell. We'll have carbohydrates present in our cell, we'll have lots of proteins present in our cell, lots of lipids, and these enzymes could actually end up breaking down our cells. So we actually need a safe way for them to be contained, so they can, once they have been produced, they can then leave the cell. So what they do, these are kept separate from cytoplasm by surrounding them with a membrane, and they can be used to digest invading cells or to break down worn out components of a cell. So obviously parts of our cell will renew, so we might have an old mitochondria that needs to be gotten rid of, so we might break them down using the lysosomes, and the cell might need to go through cell death, so the lysosomes would take over that. But we might also just have an invading cell that needs to get rid of. So they can break down materials ingested by phagocytic cells, such as white blood cells. And so when we talk about phagocytosis happening with white blood cells engulfing pathogens, lysosomes are then those digestive lysosomes are those vesicles that contain the lysozymes, which are the digestive enzymes that can then go in and start to break down all those pathogens. Lysosomes release enzymes to the outside of a cell. They digest worn out organelles, so such as mitochondria, ribosomes, and they break down cells when they have died. So when cells have gone through cell death, they then break them down, so we haven't got all this dead cells just floating around in our body. This is lysosome structure. There's not much to it. Has a membrane, double membrane, and proteins and enzymes found. Well, enzymes are proteins and found on the inside of it, and that's it really. There's nothing much else to it than that. Which leads us on to ribosomes. Ribosomes, these are found in our cytoplasm or on the rough endoplasmic reticulum. Ribosomes we draw in the past as just like a little dot. They are tiny. They are super, super tiny. They're very small and they float free in the cytoplasm or they're attached to a rough ER. Now, ribosomes are made up of proteins and RNA, and ribosomes are not surrounded by a membrane. Ribosomes are the site where proteins are made. Now, ribosomes are made up of two subunits. We have the top subunit and the bottom subunit. We call it small and large subunits. They play a hugely important role in protein synthesis. So when we come on to look at protein synthesis, ribosomes are the reasons that proteins are synthesized. And so we have two types of ribosomes. We have the 80S type, which is found in eukaryotic cells, and they are about 25 nanometers in diameter. And then the 70S type, which is prokaryotic cells, so they're smaller. So when I talked about subunits, this top half would be the large subunit, and then we've got our small subunit. We also have microvilli inside our cells. These are like finger-like productions of epithelial cells to increase surface area for absorption. And then, oops, let me pause this a second because I lost where I wanted to go with this. So the organelles, what has happened? Why has it gone here? Okay, so I don't know, I've lost a slide somewhere. So eukaryotic cells and their organelles. So we now have this list of new organelles that we need to know. Obviously, we still have our chloroplasts from the past. Chloroplasts we're not going to look into again, super detail here. There's loads of different layers to the chloroplast now at this point. They have things called like stroma and granum and lamella and thylakoid cells, thylakoid stacks in there. And basically, we look into them more in A2. So there's not really great importance of going into great detail of them right now because we look into it more later. But our chloroplasts are small flattened structures, and it's obviously our site where photosynthesis occurs. So the organelles that occur in specialized cells actually vary. So we say, oh yeah, these are all our organelles, but actually we have some variants. So in specialized cells, our organelles do actually vary. So organelles can vary in specialized cells. So in multicellular eukaryotic organisms, cells become specialized to carry out a specific function. So you'll have done lots of this at GCSE, where you'll have found out about things such as neurons, nerve cells, epithelial cells, muscle cells, egg cells, sperm cells, cardiac cells, liver cells, plant cells, root hair cells, and it's all about specialized parts of it. And so a cell's structure, and so we specifically say that a cell's structure is its shape and organelles. So structure isn't just its shape, it's its shape and organelles. So a cell's structure helps it carry out its function. So you'll have noticed this in the past before, that we'll have, for example, a red blood cell is round. It doesn't have a nucleus. It has that indentation in the middle, which is probably going to make it look like it's got a nucleus, but it's that indentation. It has a donut shape. It doesn't have a nucleus so that it can fit more oxygen inside it, so it can carry more oxygen around the body. And then we could have a neuron. This is a really badly drawn neuron, but this is a neuron with dendrites. And again, it's got different shapes to carry on its job. It's got axons, it's got myelin sheaths to help carry everything. Then we could have ciliated cells that have got the little hairs on the top. So these are all eukaryotic cells, but they've all got different shapes to them. And these different shapes, the different structures help carry out a function. So depending on the cell's job, a specialized cell can look very different to the cells that we've obviously originally seen and spoken about. So in the exam, what can actually happen is you can get asked a question where you need to apply your knowledge of organelles to explain why a specialized cell is particularly suited to its function. So what you would have to do is you would have to think about what organelles the cell needs to do its job. If the cell uses lots of energy, it would have lots of mitochondria. So whilst all the cells may have all of those components, they can have them in different varying amounts. So if it's a cell that's going to use a lot of energy, so if it's a muscle cell, for example, it's going to have tons and tons and tons and tons and tons of mitochondria in there. It's going to be full of mitochondria. Whereas if it is a cell that makes lots of protein, it's going to need lots of ribosomes. So an example of this would be our epithelial cells in our small intestine. And I'm going to actually try and plug my pen in, try and get this to work, because my drawing isn't working very well here. So what they can do in the exam is they can give you sort of any sort of specialized eukaryotic cell and go, why is this, what's different about its structure that helps it carry out its job well? And you have to sort of pick out what it can do that helps it carry out its job, what's different about it. So let's draw an epithelial cell. Let's undo that. Right. So this is going to be my badly drawn epithelial cell. There we go. Let's pop our nucleus in there. Let's pop some mitochondria in there. There we go. So, example, this is our epithelial cell. Epithelial cell in the small intestine. In the small intestine. And you could get asked, epithelial cells in the small intestine are specialized to absorb food efficiently. So then you'll go, okay, let's look at this then. Let's point out some of the different things in here that will help absorb food efficiently. first thing that you'll notice is you have those microvilli on the cell wall. So this, well, not cell wall, sorry, and the cell membrane. So the walls of the small intestine have lots of finger-like projections called villi. These increase the surface area for absorption. So let's pop that there. So you can pop on the villi, increase surface area. Then we can go, okay, what else? So the epithelial cells on the surface of the villi have folds in the cell surface membrane, and they're called microvilli. So then we can mention our microvilli. Microvilli, again, increase our cell surface area even more. Cell surface area is increased even more. What else have we got? Actually, we've got lots of mitochondria. Why would having lots of mitochondria be useful? Well, we've got lots of mitochondria. This is useful because it provides energy for the transport of the digested food molecules into the cell, which needs active transport. So it's that energy provision. So you can sort of see now that you're sort of saying what there is and why that's useful. What there is, why it's useful. Obviously, you should know multicellular eukaryotic organisms and specialized cells are grouped together, and when they group together, they form tissues. A tissue is obviously a group of cells that work together to perform a particular function, and different tissues work together to form organs. Different organs make up an organ system, and therefore organ systems make an organism. So, for example, if we use our epithelial cells, groups of epithelial cells together will group together to make epithelial tissue. Epithelial tissue, muscular tissue, glandular tissue, which secretes chemicals, all work together to form the stomach, which is an organ. The stomach is part of the digestive system. So this is an organ system made up of all the organs involved in digestion and absorption of food, such as the small intestine, large intestine, and liver. And then you can say you have all these other organ systems that together would work together to make an organism. So that is eukaryotic cells and organelles. We're now going to have a look at prokaryotic cells and their organelles. So let's go back. Hopefully hope that all my slides are here now. Prokaryotic cells. So prokaryotes. Prokaryotes are simple cells, usually, with few organelles. Bacteria such as cholera are prokaryotic. And what prokaryotes are made up of are these organelles, which we'll then look at in some more detail. They're made up of plasmids, which is small circular DNA strands, genetic material, cell surface membrane, a cell wall, a capsule, potentially a flagellum, and a mesosome, which are the sites of respiration. So, oh, it's really annoying. 1 second. I've deleted something I wanted. What have I done? That's what I want. Control C. Okay. Oh, what's happened? I'm trying to share and the whole screen's crashed. Really sorry, I don't know what's happening. Just give me a second. My computer's decided to crash on me. Quite spectacularly. Here we go. So I need to share this now. So 1 second. Share screen. So here is a diagram of the bacterium. Any prokaryotic cell would work for this, and we're just going to have a go now at actually talking about each little part of the organelles in a bit more detail. So what we're going to look at first is we're going to look at the cell wall. So the first bit we're going to look at in detail is the cell wall. I've highlighted it there for you. I'm just going to add the information up here for you. So the cell wall. Cell wall in a prokaryotic cell does exactly the same function as it would in a plant cell. The cell wall supports the cell and prevents it from changing shape. So it's support and structure. It's made of a polymer, and this is quite interesting. It's made of a polymer called murine. So it's made of a polymer called murine. Made of a polymer called, and I always have to double check how to spell this, murine. Murine is a glycoprotein. And a glycoprotein is a protein that has a carbohydrate attached. So that is our cell wall. And then what we're going to look at now is what have we got? Plasmid. Where's my plasmids? Here we go. Yeah, plasmids. So plasmids. What plasmids are? So we've said prokaryotes don't have a true nucleus. Instead, they have these things called plasmids. And plasmids are small loops of DNA. That aren't part of the main circular DNA molecule. So in the middle, we've got the chromosome. Here it says chromosome. Sometimes it's classed as circular DNA molecule. Plasmids are small loops of DNA that aren't part of the main circular DNA. So these are, let's pop plasmids there. Plasmids, small loops of DNA that are not part of the main circular DNA. What plasmids are? Plasmids contain genes for specific things. So plasmids contain genes for things like antibiotic resistance, and they can be passed actually from prokaryotic and can be passed between prokaryotes. So they can be passed from one prokaryotic organism to another. Plasmids aren't always present, and some prokaryotic cells have multiple plasmids, so it can vary. So they can be passed between prokaryotes, and some don't have them. Sometimes are present, sometimes are not present. Let's move that over there. Then we have, let's talk about the circular DNA then. That makes more sense. Let's go to the circular DNA next. Here it says chromosome, and sometimes we call it circular DNA. So it's a bit of a strange one. It sort of has multiple names. Let's call it circular DNA / chromosomes. So what this is, unlike a eukaryotic cell, prokaryotic cells do not have a nucleus. So we know that, we're happy with that. Instead, our DNA floats free in the cytoplasm. So it's free floating DNA. Free. So it's free floating DNA. It's circular DNA and it's present as one long coiled up strand, and it is not attached to any histone proteins. So it's circular and not attached to any histone proteins. Then we sometimes, again, I'm saying they sometimes have them. They sometimes have a flagellum. So flagellum. So the flagellum, let's look a bit more into that. Flagellum, plural flagella. So flagellum. Technically, this one would have flagella because it has more than one, but we'll talk about a specific one, it's just one. It is a long hair-like structure. So it's long and it's a hair-like structure. Obviously, occasionally eukaryotic cells can have these. So an example of a eukaryotic cell does have a flagella would be sperm cells, and generally they don't. Prokaryotic cells quite regularly can have flagellum. The reason they have flagellum is because prokaryotic cells are obviously unicellular organisms, and they need a way to actually be able to move. And so what these do is they rotate, these long hair-like structures, they rotate to make the organism move. So they rotate to make the prokaryotic cell move. Again, not all prokaryotic cells have flagellum, some have more than one. So not always present, sometimes more than one present, more than one is present. Then we have got the cytoplasm. Where's our cytoplasm? Here we go. Cytoplasm. Cytoplasm. So the cytoplasm of a prokaryotic cell, the cytoplasm has no membrane-bound organelles, unlike our eukaryotic cells. So that's the really, really important bit. It has no membrane-bound organelles. Make it a bit smaller so it just goes there. It has ribosomes, but they're smaller than those in a eukaryotic cell. And that's it for the cytoplasm. Nothing really else fancy about it. We also have a cell membrane, also classed as a plasma membrane sometimes. So let's just talk about the membrane here. Cell membrane, cell or plasma. It's made up of lipids and proteins, very similar to the membrane in a eukaryotic cell, and it controls the movements of substances in and out of the cell. And then finally, the only thing we haven't mentioned, and there is one on here actually, is a capsule. So again, this is something that sometimes they have, sometimes they don't. Let me just highlight that. Here we go, capsule. So some prokaryotic cells have something called a capsule. What a capsule is a capsule basically goes around the outside of this prokaryotic cell. So as you can see by the images, it's on the outside, and it's made up of slime. That is its official name. It's made up of secreted slime. Made up of secreted slime. It helps protect the bacteria from attack by cells of the immune system. So that is the purpose of it. So it helps protect bacteria from attack by cells of the immune system. So again, I'm going to pop here, not always present. So flagellum, capsules, plasmids are not always present. Everything else is always present. There is a thing called pilus, and they're sometimes present, they're sometimes not present. They're those little projections coming out of the cell. They're like mini flagella, really, but they're actually used for more of a defense to get rid of other organisms that might be coming nearby. And so that is a general overview of prokaryotic cells. Oops, let's just have a look at it from here. Let's clear all that writing so we can actually see. So yeah, this image here, we're showing that obviously we've got our 70S ribosomes, we've got our smaller ribosomes, we've got a mesosome, plasmids, flagellum, nucleoid, which is our circular DNA, our cell membrane, our cell wall, and our capsule slimy layer. Not all prokaryotic cells have all the parts that are shown here. That's really important to remember. Here's another example of a real life one. So here's cholera, which is a prokaryotic cell. So you can see that cholera has a flagellum, and you can see that it's got its circular DNA inside. Another image of cholera. So don't want cell surface membrane. Really. Managed to get these out of order, which is really not helpful. Right, I've lost the next bit that I wanted. Fractionation, diffusion, osmosis, active transport, digestion. Okay, that's really annoying. I've lost that one as well. And no, I don't want just annotate nothing. Let's do this last bit on the whiteboard then. Okay, so clear that. No, I'll just skip to the next page. So viruses. These are a weird one. GCSE, we don't really define them clearly enough. We say viruses are cells. They're not. They are not cells. Viruses are not alive. So we cannot kill viruses. And they are not cellular. So that's basically one of the most important things about viruses. So viruses are acellular. So they are not cells and they are not alive. So that's really important. So viruses, what are they then? Well, they're nucleic acids. Viruses are made up of nucleic acids surrounded by proteins. So they're nucleic acids that are surrounded by proteins. They are not alive either, which is really important to remember. Lots of people fall into the traps. They're not cells, and let's pop over here, not alive. People often fall into a trap when writing about pathogens, saying, oh, bacteria is killed. Yeah, we can say bacteria is killed, bacteria is destroyed. And then they say virus is killed. Well, no, we can't kill a virus because the virus isn't alive. We can say that cells die, and we can say that cells could be killed because cells are living, but viruses are not living and they are not cells. And that's really, really important to remember. They are smaller than bacteria, which is probably something that you can remember from GCSE. They are super tiny. So they are smaller than bacteria. Obviously, bacteria are tiny and smaller. These are even tinier. So they're smaller than bacteria. And so for an example, a HIV virus is about 0.1 micrometers wide in diameter. Unlike bacteria, they have no plasma membrane, no cytoplasm, no ribosomes. Not surprising because they are acellular. They are not cells, so they are not going to have a membrane. They're not going to have a cytoplasm. They're not going to have ribosomes because they aren't a cell. All viruses, though, invade and reproduce inside cells. So this is their way of working. So it's really important to realise that all viruses, despite where and how they work, so we might have retroviruses and different types of viruses, all viruses invade and replicate inside cells, invade cells and reproduce inside them. These cells that they replicate inside of are then called the host cells. The only other thing is we need to just know a little bit about what an average virus looks like. Viruses look so different. They can all be really, really different. So you can get some viruses that look like this. And we've got there, basically they contain the genetic material on the top and a bit like this. And they come along and they'll sit on top of the cell and they inject their genetic information in. You can have other ones that look a bit more like this, and I'm going to try and draw it badly. And then again, inside it, you've got your DNA. So in this, well, RNA, sorry. So, or DNA, it depends. So viruses contain a core of genetic material. So again, this is our core of genetic material. So that's our genetic material. It can be DNA, it can be RNA. So that's our core of genetic material. They have a protein coat, which is around the core, and this is called the capsid. So this is the capsid here on this one, capsid. This is the capsid on this one, and that's that protein coat that's around the core. And some have attachment proteins. Well, they do all have attachment proteins. This one has attachment proteins at the bottom. This one has attachment proteins at the side. So let's label them together, actually. Attachment proteins. These let the viruses cling on to a suitable host. So basically what happens is virus can cling on to a host cell. Obviously, the virus won't be that big in comparison to the size of the cell. The cell will be massive, but I haven't really drawn it very well. So actually, it would probably look more like this. That's the outside of the cell. That's the virus clinging onto it. DNA and RNA are nucleic acids. So actually, when you look at this, that's all viruses. And virus is just genetic material that's coated in some proteins that help keep it safe and then help it attach to a cell. So next biology session, what we will look at is how prokaryotic cells replicate. So obviously they replicate by binary fission. So we'll do a bit of a recap of binary fission. And then we'll have a look at analysing cell components, the different components of cells, and how we might analyse them and look into microscopy in a little bit more detail, and then go into mitosis as well. Tomorrow